Okay guys, this is the first of two videos that'll walk you through what is a rather short presentation on bacterial transformation. This is the subject of the lab we're doing with this unit, the PGLO lab, but it's also a concept that you have to know about for the AP exam, and it's really one of the only concepts from bacteriology that you need to know. So we'll give you kind of a crash course now. We'll talk about this just a little bit toward the end of the year as well when we talk about the human immune system. But the big key here is we want to figure out what bacterial transformation is, and we also want to figure out how can we actually use this to benefit us. So you'll notice on that first slide I had, this is sort of the roots or foundations of biotechnology. You've seen part of biotechnology presentation at this point, and that stuff's pretty advanced and complicated. But before we could get to that point, scientists had to realize that DNA was the genetic material, and that genetic material, passed down traits from one generation to the next. And so there were several scientists that played a hand in this. Now we're going to start with a date and place you do not need to know. We're going to start in England in 1928 with a scientist named Frederick Griffith, who you saw pictured with his dog friend on the first slide. And you might recognize Griffith's name from freshman year. It's one of the scientists you have to learn in that class. And Griffith was a bacteriologist who studied disease and was sort of in a race with some other scientists to develop a vaccine against pneumonia, because pneumonia still today is quite a lethal infection. Today it's more of an issue in older people than it is in somebody your age, but can be a very, very serious infection that can lead to death. And so they sought to cure it. Now, Griffith had identified two different strains or two different types of the primary bacterium that causes pneumonia, which is Streptococcus pneumoniae. And you see here in this picture, they've got a plate where they're culturing some Streptococcus. Each one of these little colonies would be about 1 million bacteria. And what Griffith had noticed is that one of the strains, which he identified as the S strain, right here, appeared to be pathogenic, or in other words, would actually cause disease. The other strain he identified, the R strain, appeared to be non-pathogenic, meaning that it would not cause a lethal disease in an organism that had contracted it. Now, Griffith used laboratory mice to test whether or not a bacterium was pathogenic or not. So in the case of strep pneumoniae, he set up an experiment where he had a control set of mice where he did not inject them with anything, and obviously he would just observe these for their normal behavior. And then he had 4 other experimental groups. His first group he inoculated with a living S bacterial injection, so again a pathogenic one. And so of course expectations for this pathogenic mouse, let's draw a little mouse head like this. I don't really know how to do this. There we go. Expectations for this mouse, it would die, right? Because we infected it with pathogenic bacteria. The second experimental group would involve inoculation with the R, or the non-pathogenic, the harmless bacteria. And so we would expect that these mice would live, right? The third group, he would infect with a heat-killed S. So a group of bacteria that are dead, they were pathogenic when they were alive, but now he has killed them. The idea here sort of is twofold. One, we want to know what is it about the bacteria that causes disease. So does the bacterium have to be alive to make you sick, or would something on its surface or something like that make you sick? The second idea here is just in terms of how vaccinations work. And I know that you guys don't have a great deal of background in that, but some vaccines are live bacteria or viruses, some vaccines are dead. And so we need to know how will it affect the body when that bacterium or virus is alive versus dead. And so that was kind of his reasoning for this third one. Now again, the expectation probably is that these bacteria will live, right? Or excuse me, these mice will live, because the bacterium was a bad one, but now it is a dead bad one. Then the fourth group was a mixture of heat-killed pathogenic bacteria and living harmless bacteria, which again should both be harmless at this point, right? Living bacterium that is not pathogenic, and then some dead bodies of a bacterium that used to be. So again, we would expect that these mice probably also would live. Now what's interesting is that the results Griffith got did not match up with these hypothetical results. So here are our 4 experimental groups again. When we, and notice too, I'm calling them all experimental groups. Your textbook calls these controls. It doesn't really matter how you look at it. But the living S cells, the living pathogenic bacteria, did what we expected, right? Mouse dead. The living harmless bacteria, the R cells, did what we expected. Mouse healthy. The heat-killed S, dead pathogenic, did what we expected. The mice survive. Those dead bacteria don't really affect you. But check out this last group. Right? A mixture of dead harmful ones and living harmless ones actually killed the mouse. And what's more is that when he performed a little Mickey Mouse autopsy, he was able to culture living pathogenic cells from the lungs of these mice. So one of 2 things was going on. Right? The pathogenic S cells either had some way to resurrect, right, come back to life, or the harmless R cells learned how to be bad, learned how to be pathogenic, and killed these mice. Now, Griffith went with the second option. He did not expect that something would bring the pathogenic bacteria back to life, so there must have been some transfer of information between the dead S cells and the living R cells. Here's my sweet Transformers background. So several other scientists played a part in determining that it was DNA that was that genetic material that would transfer between the cells. Griffith suggested that, but he was much more interested again in the pathogenicity here. He's much more interested in the immunology side of things. And so a few other scientists got involved from different parts of the world too, in determining very clearly that DNA is what was actually being transferred. The scientists that you had to learn about as freshmen and that you might still recognize their names are Hershey and Chase. Hershey and Chase is fun. It's Alfred Hershey and Martha Chase, so there's a female involved with that experiment, and they determined very clearly that DNA was the genetic material. Also, a little guy named Oswald Avery supported this with a lot of good evidence. But Griffith is credited and famous for his discovery of bacterial transformation. The concept that bacteria can change their genotype and phenotype, right? Their traits and their specific genes they carry by assimilating external DNA from the environment. A crazy concept, but really interesting, and something that is unique to bacteria. So how do they do it? The way bacteria do this, and what makes them different than us as eukaryotes, is that they carry extranuclear DNA. I think we might have mentioned this way back at the beginning of the year. But unlike our cells, bacteria have a single circular chromosome to start with, but then they also have these small loops of double-stranded DNA called plasmids. And these little loops, these little rings, are found outside of the standard genome, so outside of the normal genes of the bacterium. And these plasmids can carry several different genes. The plasmids also can be self-replicating, meaning that they will use the cell machinery to make copies of themselves, or they can be spliced into the cell's standard genome and then copied when that bacterium copies all of its DNA. And then you would kind of splice the plasmid back out again. Now, what makes plasmids most unique is that they can move from one cell to another. So, in Griffith's experiment, he observed transformation, where those harmless cells picked up a plasmid from the harmful cells and basically inherited the genes on how to be bad, right? How to be pathogenic. And if they incorporated those genes into their genome, they then had those characteristics of a pathogenic bacteria. In this case, meaning they could make a certain toxin or something that would be deadly to the cells of the host. And they essentially transformed their phenotype to become pathogenic. Now, there's two ways that bacteria can do this. The first one is probably what, we're pretty sure is what Griffith observed, which is that living bacteria can absorb plasmids from the environment. So when he put those dead S cells in that mix, they probably kind of broke open, all their parts were loose there in the solution, and their plasmids were floating around. And those living R cells would have surface receptors, right, that would recognize DNA from a species much like themselves and absorb those plasmids. Now, the secondary option, if you have two living bacteria, is called conjugation, which is what you see in this picture here. Conjugation is where the bacteria transfer a DNA plasmid from one cell to the other cell very purposefully. And so I put on here, I'm kind of being silly, but it's kind of like sex, right? The one bacterium forms this bridge called a pilus with another bacterium, and they're able to pass their plasmid over to, or really they pass a copy of their plasmid over to the other bacterium. It's almost a way for them to create genetic variation. It's a way for them to share genetic information with each other. So here's a picture of how this can occur. You don't have to know specifically that this is the F plasmid. It's just the one that your textbook uses. But if you look along the top, we have an F positive cell, meaning a cell that carries this F plasmid. And it will conjugate or form this pilus with an F negative cell, right? A cell that doesn't have that plasmid to start with. So then what happens here in the second picture is the F positive cell makes a copy of its plasmid and transfers that copy over to the F negative cell. Makes its way in here, and now in the end we end up with two F positive cells. Now, in either case, like it says here on the slide, bacteria can replace the alleles they have with new alleles. They can essentially learn new skills genetically. They can start to transcribe a new protein that they didn't have the instructions for before. This is wild because no other organisms can do this, right? This is why we talk about humans, and you have a certain set of genes that you get when you're born, and those are your genes for your entire life. It's why genetic diseases are not treatable because or I shouldn't say treatable, they aren't curable because you're stuck with the genes you have. Bacteria aren't. Right? Bacteria can get new genes and can transcribe those and form new proteins. Now, the benefits of transformation depend on whose point of view you're looking at this from. Whether you're looking at it from a bacterial point of view or whether you're looking at it from a human point of view. So the second video is going to discuss why transformation is so good for bacteria, but also why transformation could be good for us.